Let's start my presentation in this difficult online session. It will be about the seasonal dynamics of microbial communities in forest soils. Perhaps not everyone considers the microbes as a component of the forest, but I would like to show how important they are and how they reflect the seasonality of the temperate forests. The work was pursued by the members of the Laboratory of Environmental Microbiology of the Institute of Microbiology here in Prague. There are several people which were coordinated by my postdoc Zander Human, and you can see the picture of our group at the wedding. And there were several collaborators that helped us to solve some parts of the work that I will be presenting, from Iowa State, Oslo University, INRA, Nancy, and US user facilities that helped us to get some data. Hello, everyone. Let's start my presentation in this difficult online session. It will be about the seasonal dynamics of microbial communities in forest soils. Perhaps not everyone considers the microbes as a component of the forest, but I would like to show how important they are and how they reflect the seasonality of the temperate forests. The work was pursued by the members of the Laboratory of Environmental Microbiology of the Institute of Microbiology here in Prague. There are several people which were coordinated by my postdoc Zander Human, and you can see the picture of our group at the wedding. And there were several collaborators that helped us to solve some parts of the work that I will be presenting, from Iowa State, Oslo University, INRA, Nancy, and US user facilities that helped us to get some data. Talking about ecology of forest topsoil from the viewpoint of microbes, we can divide the nutrients into leaf and root litter, the major sources of organic matter in soils, composed mainly of biopolymers like cellulose, lignin, and any other complex stuff. And for that, a numerous enzymes are needed to be degraded. Then we have a simple carbon-rich nutrients like root exudates that are produced by the tree roots and either given directly to their symbiotic fungi or made available to the soil microbes. And the microorganisms reflect somehow the availability of these resources. So there are saprotrophic fungi, degraders, that obtain carbon mostly by degrading organic matter, produce multiple extracellular enzymes to be able to do it efficiently. Then there are mycorrhizal fungi that specialize at the exchange with their plant hosts and get the nutrients from them. In return, they give their plant hosts access to other nutrients. They have some saprotrophic abilities, but these are variable and limited. And then there are bacteria that mostly prefer to utilize easy compounds, but some of them also have some ability to degrade biopolymers. If we speak about temperate forests, they typically experience seasonality. So if we look at the late vegetation season in September, is the time when the CO2 fixation goes very rapidly. The CO2 is fixed and goes through the roots into the soil, and then it is handed over to mycorrhizal fungi and soil microbes. The mycorrhizal fungi have unlimited access to carbon and are very active, and the respiration is high. However, in winter, the trees can't photosynthesize, so this flow of easy carbon is stopped, and it's the time when the saprotrophic microbes take over, and the activity of mycorrhizal fungi is expected to decline, and the saprotrophic fungi and bacteria should increase. But in the same time, the total production of the system is much lower. Sometimes the soil is covered by snow, but still it's very, very well living if you uncover the snow and smell, you can see you can smell the fungi there. So they are active all time, even under this snow cover, because the typical temperate forest soils never freeze. So the winter should see some activity of the mycorrhizal fungi. And to address this issue, we tried to sample what happens in soil in different seasons, in spring, summer, early and late winter. We isolated dominated bacteria and dominant fungi to have some idea about their genome composition, but we also sampled separately different topsoil habitats, the plant litter that contains recalcitrant carbon, the soil, but also the rhizosphere at the interface of the root and the soil. And we also analyzed the spruce roots. What we did is that we mostly relied on metatranscriptomics to see the genes expressed at each season and how different are the genes that are utilized in different seasons and how different are the microbes that live there in different seasons. So the rhizosphere should be the mediator actually of compound and information flow from the trees to the soil and from the soil to the trees. So it's in many cases different than the bulk soil. So if we compare the bulk soil with the rhizospheres, you can see that the fungal biomass is typically higher than the bulk soil, both in summer and winter. So it's a hotspot of fungal activity, mostly of mycorrhizal fungi. And also it's a hotspot of enzymatic activity because it offers more nutrients, and more nutrients means more energy to generate enzymes and to turn over organic matter. So the rhizosphere indeed is a valid habitat, which is in many ways different from the soil. And coming back to the experiment, we selected the 4 periods of the year. You can see that in the vegetation season, the temperature is varying, but in our temperate spruce forest, it's around 12 degrees most of the summer season. And in winter, the temperature in the soil drops to roughly 0. It's very constant because it's insulated by snow, and there is a stable environment when the soil is not frozen and the microbes can live, but they should cope with the limited access to nutrients from the trees. So if we look at the community as it is, and look at the potential, which is expressed as a metagenome, as all the genes that we can recover from the soil, we can see that litter DNA, these green dots, is different from soil DNA, which means that there are different microbes in each of them. But if we look at the full and open circles, they are mixed together. The full circles are summer, the open circles are winter, and there is no seasonality in the genomic information. There are the same communities in summer and in winter, different between litter and soil, but not different between seasons. If we look at the activity, the genes that are expressed, we can see again the separation of litter in green and soil in brown. But in addition, we can see the separation of summer samples with the full symbols and winter samples in litter, and summer samples and winter samples in soil. So the microbes are the same, but they express different genes in the different seasons to cope with the conditions that they are there. And one of the target processes that can be altered because of this alteration of carbon flow is the carbon cycling. And we can analyze them by addressing the genes for carbohydrate utilization in forest soil. There are many different families of genes that attack different targets. The targets can be divided based on their presence to plant cell wall polymers, fungal cell wall polymers, some cell wall polymers of bacteria. And if we understand which genes are expressed, we can see which compounds are utilized each part of the year. We can look at who is producing which enzyme. So here in this graph on the x-axis are the most abundant enzymes that turn over carbon, and with the colored bars, there is the breakdown of community of the decomposers. The red color is fungi. You can see that many genes are mainly expressed by fungi. The blue color is bacteria, and there are some other colors, like, for example, the green for plants. So the upper picture shows the share of the total transcription, and are the most transcribed genes. If we look at the DNA pool at the metagenome, this is the bottom picture, and it has the same ordering. You can see that sometimes there is a big share of metagenome in the bacterial cells, only very little is expressed. We can also divide the enzymes by their targets. So, for example, the enzymes highlighted in green, like cellulose, you can see that the second, third, and fourth most abundant enzyme is at cellulase, actually. So it really demonstrates that the expression of cellulases and the utilization of cellulose is very important. But you can also see the yellow color is the most abundant enzyme. It is actually alpha-glucosidase, and its activity is towards reserve compounds like starch or glycogen, the compounds that the microbes can store in summer and utilize in winter when they don't have other resources. So you can see that this enzyme is very important. We can also ask how many genes and how many microbes are able to run the processes, and this is a list of some of the enzymes that we can follow. And you can see that in the DNA, you have hundreds to thousands of genes, individual genes that you can record there, which means that there are really hundreds to thousands of microbes that are able to catalyze the same process. It's not that it is one single microbe that is dominating the process. It's really a cooperative task. If you look at the expression, again, it's tens, hundreds, sometimes thousands of microbes that do the same process, and jointly they perform the decomposition of organic matter. We can later look to the genes that are overexpressed or underexpressed. One season, you can see the dashed lines. Everything which is above the dashed lines are genes that are more increased in summer, which the ones that are below the lower dashed lines, it's genes that are increased in winter. And you can see the green and brown dots. The green dots are the genes that are over or underexpressed in litter, and the brown are in soil. So you can see that there are very many enzymes that are overexpressed in soil in summer, and this should reflect the higher level of seasonality in the summer season for the roots. And what is it about? If we look in the soil specifically, where the seasonality was most prominent, you can see that there is much higher share of enzymes attacking starch and glycogen, the reserve compounds, and also trehalose, the reserve compound of fungi, that are produced in winter, because the winter needs to call for this energy reserves for the microbes to survive the lack of easy carbon from tree roots. If we go to cell wall turnover, you can see that the summers are slightly higher than the winters, both in the litter and in soil. For bacteria, it's rather the litter. And this should indicate that perhaps the growth and regrowth of their cell structures is higher in the summer, where there is more energy and more carbon flowing from the trees. If we look at lignin and cellulose in soil, we can also see that they are more attacked in summer than in winter, most of these compounds, which is a little bit counterintuitive. One would say that if there is an easy carbon, the microbe should preferentially use this. But we believe that this is actually a proof of priming, the fact that this easy carbon can initiate the degradation of organic matter by the enzymes. So it seems that the activity of trees not only gives the possibility for faster growth, but it can also initiate faster decomposition of recalcitrant carbon compounds like plant debris due to priming and due to higher activity of lignolytic and cellulosic enzymes. We can also look at the decomposition as a gross process and compare it between seasons. Here with the different colors are different seasons. You can see December to March, March to June, to September and September to December. And you can see that December to March is very low temperature, around 1 degree centigrade. Then March to June is 5 degrees average temperature. Summer is relatively hot with 13 degrees average temperature. And again, the autumn is 6 degrees. But you can see that actually the loss of dry mass of spruce needles or grass litter or even cellulose is relatively fast in each of the seasons. So the decomposition never stops. But if you look at the more recalcitrant compounds like cellulose, you can see that it's very fast. Its decomposition is very fast in summer and much slower in the winter seasons, which again falls to the fact that when the trees are active, the decomposition is much more easy and perhaps also mediated by the higher temperatures. So the summer is the time when the cellulose and other recalcitrant compounds are rapidly degraded, while the winter is slow in that. There are also other genes that show seasonality. One example can be nitrogen cycling processes like nitrification, dropping in early summer, or ammonia assimilation, which is restricted to early winter to early summer, and other processes. So it's not only carbon cycling that shows some seasonality. And there would be a plenty of processes. For example, the winter sees very high nitrification, the energetic use of nitrogen, so to say, while the summer doesn't need this source of energy because probably it's generated from the easy carbon that is delivered by tree roots. The other view that we can get of the system is through metabolomics. So we can prepare soil, extract metabolite, filtrate and isolate them. And then after derivatization to identify individual compounds and compare their abundance. And you can clearly see that the compartments in different colors, roots, rhizosphere, litter, and soil are distinct. They have distinct metabolites. Most distinct are the roots. And within each habitat, the seasons are again distinct. So there is specificity of the metabolites to each habitat, but also habitat seasonality. And then looking at the metabolome seasonality, for example, comparing just the summer and winter, we can see that the empty symbols are distantly located from the closed one, which tells that the summer and winter in this case are different in the metabolome profiles. So it's different metabolites that are available to the microbes, and they somehow adjust the metabolism to that. One example can be this chenic acid, a phenolic acid, which is very, very high in early summer, both in rhizosphere and soil, but particularly in rhizosphere, drops to less than 1/4 in the winter. So it's probably some signal which has some role in the seasonality. We can also look at the individual microbes and who is doing what. So the microbial community, although it processes carbon, stores it, decomposes and so on, doesn't work as an entity. The microbes don't collaborate with a defined goal to decompose something. No, each of them is living their own lives, searching for their nutrients and performing their own processes to survive, to double, to grow. So the correct view to understand the microbe is actually through individuals. And what we can do is to isolate the dominant taxa. This is what we did. We isolated some 20 dominant taxa from the soil that we studied. Some of them were new species even for science. So with these microbes isolated, we can sequence their genomes and identify their features. So if we look at the genomic potential, we can see that there are bacteria groups that are very rich in genes for degradation of cellulose, hemicellulose, and chitin. For example, the Acidobacteria in this upper clade, and Bacteroidetes in this lower clade. And we can even confirm it in laboratory cultures. So these Acidobacteria, which have more genes for decomposition, also produce higher amounts of enzymes in cultures. And to some degree, this is also valid for Bacteroidetes, where some of them are active in enzyme production and also rich, have genomes rich in the genes. If we have the genomes, we can go further, look back to the transcriptomes from the soil and see what do the individuals do there. And what we can see is that the individuals, most of them are able to adjust their physiology to the soil horizon, the litter or organic soil, because the conditions in each of them are different, so they need to express different genes. But many of them also show individual seasonality. So the same microbe uses different resources in summer and different resources in winter. We can also look how the genomic potential that we see encoded in their DNA is expressed. And indeed, if we follow the transcription of the taxa in soil, we can see the decomposers really produce more enzymes, the transcriptome. So it's not only presence in genomes, it's activity, is there active transcription. And on the other hand, we can see some opportunists where the genes are very rare. And indeed, even these rare genes are not even very much expressed. So we can somehow distinguish the bacteria that can decompose the organic matter. And these are more likely to be active all the year round because they aren't so much dependent on the easy carbon provided by plant roots. While the opportunists require the easy carbon, they cannot cope with the complex biopolymers, and they should be enriched in summer where the available carbon is present. So we can distinguish these two guilds. And indeed, if we look at their occurrence, opportunistic guild is super abundant and super active in summer, many times higher than in winter, because there's easy carbon there and they can live with that. While the decomposer guild is almost equally abundant all year round, maybe a little bit more dominating in winter, but this is because the opportunistic guild is so much suppressed. So the decomposer guild can easily cope in the winter times when they need to utilize this complex compounds that are their resource for the winter time. And indeed, if we look at the decomposer guild, we can even see some seasonality. They are able to switch to easy carbon in summer, but they are able to grow on recalcitrant one in winter. For the opportunistic guild, it's even more pronounced. They are striving in winter. They are trying to survive. They are trying to preserve their DNA, while in summer they are really actively growing, doubling, and metabolizing the easy compounds like organic acids or monosaccharides that are present there. Thanks to the Joint Genome Institute, we also have the genomes of some fungi, ectomycorrhizal fungi that actually mediate the transfer of nutrients from the trees to the soil. We have the genome of Amanita rubescens, Boletus edulis, Drusilla emetica, and Drusilla ochroleuca, which are dominants in our forest. And with that, again, we can ask about their expression in the different seasons. So we can compare their activity in summer and winter. And we can see that really some of them are more active in some of the seasons than in the others, like the Amanita and the Boletus seem to be more expressing in winter, probably restructuring their mycelia and perhaps relying on the nutrients provided by the tree roots of their hosts. The Russulas seem to be more active in summer because they are trying to explore higher amounts of soil and to extend in this time of the year. And we can also look at the individual fungi. This is the example of Drusilla ochroleuca, the most dominant fungus in our system. And you can see that they can also produce different genes in different habitats, but also show some seasonality. For example, in litter, when they are fruiting, they express very different genes than in litter, which just in winter it's colonized and used to retrieve the nutrients that is required to maintain the mycelia. So as a summary, forest ecosystems are seasonally dynamic in temperate forests. The temperature-driven seasonality in temperate forests affects vegetation activity, mostly the activity of trees, and ultimately microbiome activity. The decomposition rates also show seasonal variation, although we can see decomposition all year round. The microbial carbon utilization shows some seasonal patterns. the microbes pick the compounds that are present and the ones that are most attractive at this time of the year. And this is probably mediated or at least signaled by the changes of metabolome composition. In bacteria, we can distinguish opportunists that just wait for a simple carbon, and when it is not there, they just try to survive this harsh environment, and decomposers that are able to use the simple compounds, but they are also able to use recalcitrant biopolymers of plant or fungal origin and decompose them. So they are very well suited to grow in winter and recycle the organic matter in this time of the year. And the transcription then varies among seasons in bacteria as well as in ectomycorrhiza fungi and also spruce roots, because this reflects the properties of the system in the different seasons. So with that, we have seen that the microbes live very diverse life as we do our summer holidays at the sea in summer and go skiing in winter. So the same it is for microbes. It's rather about survival and nutrients, and they pick different compounds and pick different lifestyles in the two seasons. Thank you very much for your attention.
Peter, thank you very much for this fantastic insight into the functional diversity of forest soils. I think I will continue with one question for you. Actually, it's more like a practical curiosity from my side. What would be time-wise the dense enough sampling really to catch the changes in the functional of the, let's say, the microbiome in general? Do you think that once per season is enough, or would you go every day or every week, every month?
This is a difficult question. Actually, our research in seasonality was just started with some community surveys some years ago. We happened to sample 3 times a year the source of temperate forests. And there we saw that the seasons are, the samples from the same season from different years are closer to each other than the samples from different seasons the same year. So with that, I would say that the seasons like spring, summer, winter are defined easily enough to see that. However, I can imagine in temperate forests that shed the leaves that there can be some more abrupt break, and there is some observations that, for example, during the bud break and the development of new roots in the spring, there is also some dramatic change. So yes, it would be nice to have once the community sampled every week and then to decide, but actually I think that no one knows. We are ourselves not sure whether we will see something really very consistent if we sample in different years. But at the moment, the techniques are so complicated and so expensive that we cannot afford it. So we hope to see a little bit. For the winter, I think it's quite simple because the system is very similar all time. It has the same temperature, there is no light. The trees are inactive. So I believe that the winter is well represented. The active seasons are probably more dynamic, and you can imagine the dry conditions, the wet conditions. If there is a precipitation, there is a flush of activity. So there are very many aspects, and seasonality is just one of them.
Okay, thank you very much. I'm slightly confused, to be honest, not with you, but we have a waving hand here by one of our participants. I'm not so sure, is it a question or just a congratulation? Justinia? Well, I have got no response from her, so I guess it's a congratulation. And I think we should all congratulate you for this fantastic presentation. Oh, sorry, we have a question. Yes, by Douglas Godbold. Douglas, please.
Oh, it's dark.
Yes, you're online.
Yeah, I have a question. Very excellent talk. Actually, 2 questions. One is you get this increase in trihalose in winter, right?
Yes.
Trihalose is a well-known cryoprotectant. So I wonder if you could that. And the other thing is the difference you get between Boletus and Amanita in winter compared to Russula could be because Boletus and Amanita actually form a lot of hyphae, whereas Russula doesn't. Right. So have you got a comment on that? It's actually the activity of the hyphae that have to survive over winter. Do you think that's
yes? I think that we have some links between this and the exploratory styles of the fungi, that we may perhaps see that the ones with the same exploratory type seem to react the same way. There are some fungi that really explore the environment very widely, and for sure they are in a different situation if they want to maintain their network in winters. First, we thought that the mycorrhiza renews their network every year, and the mycelium dies back, and then it's reproduced. But it seems that it rather makes sense for them to exchange the carbohydrates with the tree roots and to maintain it, because then it is more efficient, like economically efficient, not to regrow. So obviously the mycelia that are very much stick to the roots are in a different shape than the ones that need some transport into the soil. So I think this can be some breaking point, but in fact there are too few observations. We're trying to get another genomes of ectomycorrhiza fungi and another transcriptal mappings. That is too little to say that there is a general rule. This is, at the moment, it's just an observation.
Thank you. Very good.
Those were both questions? Yes. Okay, Douglas, thank you very much for your question. I think we have no one else for discussion at the moment, so I would like to thank again to Peter for the presentation.
Thank you for the opportunity. It was very nice. Thank you. Good luck.
And we will continue with, how could I say, the presentation of the group of our laboratory on the ectomycorrhizal studies. And I will be the one who will give this presentation. It's a poster presentation, so it's supposed to be a very short one. The title is complicated because I try to join in basically everything that we are doing in terms of ectomycorrhiza functional and taxonomic diversity in the lab. But I will just present a few of the papers that we have published recently. So basically, we know that the soils are a significant reservoir of the global biodiversity and also functions that contribute to many crucial ecosystem functions, basically. And also the previous speakers on the ectomycorrhiza and soil microbiomes basically struggle in the same direction to disclose this highly complicated system called the forest soils. So one study that actually is not published yet, but it basically compared different time-wise, different stages of the beech and silver fir seedlings, indicated that between the early stage, or let's say the seedlings are immediately after the planting, and the ectomycorrhizal community 1 year after planting differs completely. So basically the community in the same seedling can change almost completely within 1 year after changing the soil or changing the system where they are growing it. The easiest way, the first study was made with a description of ectomycorrhizal basic morphotyping, so manually, which is very time demanding. So all the other studies or the recent studies we do now with the NGS as much as we can. So we don't follow just ectomycorrhizal fungi, but fungi in general, and also archaea and bacteria. And using this approach, Tina Unuk made her PhD on the silver fir ectomycorrhiza, where she compared young and adult trees from two different phenological groups. And what she found out is basically that the young silver fir trees from the same stand differ according to their flushing time. So if they're an early flushers, the ectomycorrhizal community will differ from the late flushers. Of course, this difference disappears when she was sampling the old, let's say the mature trees, so from the same site as well. So the second study was already briefly introduced by Douglas, Basically, it's a whole Europe study that included 140 different sites, and where the ectomycorrhiza was studied from 4 different tree species, I think. It included all the many researchers from European countries, including our institute, our small contribution of 2 sites. And as explained before, the host and the soil, basic soil characters are the main drivers in the let's say, diversification or separation of different communities in ectomycorrhizae. The last and the recently published paper was also a kind of large-scale study. In this case, it was not a field study, but just a literature review, where about 100,000 papers dealing with soil biodiversity in general and ecosystem services functions were basically data mined. And the 2 key things that we can say based on this study is that, first of all, the Earth, or let's say the terrestrial ecosystems, are definitely undersampled. There's a huge concentration of studies focusing on Europe, on North America, and some points in South America and Asia, but most of the soil or terrestrial ecosystems are actually still remain undersampled or completely undisclosed. Unfortunately, I didn't put on the graph where we covered the biodiversity studies and the ecosystem function studies. And the fact is that basically, if there is a soil biodiversity study, it more or less never includes also any functional study, or vice versa. So the coverage in this 11,000 and something reviewed paper was just 0.6%. So basically, several hundreds of papers really included both soil biodiversity study, regardless to the taxonomic group, and the function of those groups in the soil. It's a poster presentation, so I'm supposed to be short, and I hope I did not go due over time. So at the end, I would just like to acknowledge the 2 main financiers of this let's say, compendium presentation. This is the Leibgen-Mon project and the program research group Forest Biology, Ecology and Technology at the Slovenian Forestry Institute. And then, of course, there was a bunch of other projects, national or international, which financed each of the shown results. Thank you very much. Okay. Okay, so obviously there were some technical problems with the Zoom, with the people, the participants on the Zoom. Okay, let's continue. The next poster out of the 7 poster sessions in this early afternoon session will be presented by Tanja Mrak. She was also working on ectomycorrhiza. This time it's an ectomycorrhizal fungi on Quercus pubescens that are suffering from drought and wildfires. So Tanja, please burn and dry the ectomycorrhiza fungi for us.
Hello, everybody. I believe that all of you present here are concerned about our forests in the future. Heat waves, drought, wildfires. How will they affect complex and interconnected below-ground environment? To shed light on this perspective, we investigated vitality and community composition of ectomycorrhizal fungi of Quercus pubescens in a natural setting of Slovenian sub-Mediterranean. In 2 years of study, the area was affected by summer drought in 2016 and 2017, and the superficial wildfire in August 2016. We sampled topsoil ectomycorrhizal roots from 2 bedrocks, flysch and limestone, where soils with different water holding capacity are developed. we observed that the ratio of vital to non-vital mycorrhizal roots strongly decreased due to stress events in 2016 and 2017. Community composition of ectomycorrhizal fungi was characterized by high abundance of Tomentilla and other Telephoraceae, Sebaceae, and stress-resistant Xenococcum. Ectomycorrhizal community composition was mainly affected by the plot, but was not related to different water holding capacity of soils at the plots. However, different water holding capacity of soils affected the abundance of stress-resistant Xenococcum. Wildfire induced a significant increase in Xenococcum abundance and a significant decrease in Sebaceanales. The most common in the area were ectomycorrhizal fungi with abundant emanating hyphae and no rhizomorphs, so-called short distance exploration type. This type had the highest abundance at the plot, which was most affected by the wildfire. The presented results indicate a complex interplay of abiotic stress factors, soil type, tree vitality, and ectomycorrhizal fungi that needs to be considered. Thank you for your attention. Thank you.
So if we have no discussion, we will continue with the next presentation. The next two poster presentations will be given by Tijana Martinovic. The first one is entitled Describing Succession of Decomposer Fungi on Coarse Wood Debris in Virgin Forest Reichenauski Rock in Slovenia. Tijana, please.
Hello, everyone. My name is Tijana, and today I will talk about the succession of decomposer fungi on coarse woody debris in Virgin Forest Reichenauski Rog in Slovenia. This is a part of my collaboration with the Slovenian Forestry Institute. As this is probably one of the most described forests in Slovenia, we have many previous records that already showed a change in community composition of fungi during different decomposition stages. However, different methods were used, so our main focus was on introducing next generation sequencing as a new method for us that would complement these previous records or even become a new standard for the microbial community survey in this forest. So we selected a small subsample from 5 stages of decomposition of beech wood, one being the earliest decomposition stage and at 3 different depths. However, I have to point out here that we aimed at deeper sequencing, but at the expense of biological replicates. So we do not have biological replicates for different depths. So here, at the first glimpse of the results, we are looking at the decomposition stages only. And on this NMDS graph, we can already see that there is an indication of different communities associated with different decomposition stages shown in different colors here. If we move to the right side, we can see that there are some differences in the Shannon diversity index. From the first to the last or the 5th, so to say, decomposition stage, but we didn't find any significant differences when looking into the taxa richness. Now, if we look into the relative abundance of fungal taxa, we can see that the early decomposition stages were characterized by higher relative abundance of Basidiomycota compared to Ascomycota, which changes in the later stages of the decomposition. If we focus on the most abundant fungal taxa, we can see that, for example, our Basidium showed in light blue here, or Purpura oxalium showed in light green are present in all the decomposition stages, while Bierkandera, composing of many white rod species, was more dominant or more abundant in the first decomposition in the first decomposition stage. So altogether, we got a first look into how the fungal communities look like in these 5 different stages of wood decomposition. We have shown that the succession is not unidimensional, but mainly we have shown that our results complement previous records and offer new insights in both understanding the succession of fungal communities in this decomposing wood, but also we brought new understanding of the limits of different methods. With that, I would like to thank you for your attention, and I'll be ready to take any questions. Please feel free to contact me through my email or my social media handles. Thank you.
So since Tiana was very fast with her first presentation, I would have a question for her. And I would like actually kind of hypothesize from your side. Do you think that the differences between Asca and basidiomycetes in terms of the decomposition level could be a function of the complexity or simplicity of the substrate available?
Yes, I would. Yes, thank you, Tina. I would most definitely think so. This would be probably best studied on a bit larger sample, I would say. As I mentioned, we lacked some biological replicates. So this is a preliminary view on how the fungal community composition looks like in these decomposition stages. And considering that the 5th stage of decomposition most probably has very different carbon sources available, I would say that yes, this could go well with this change that we see in Ascomycota versus Basidiomycota.
Okay, thank you very much. We will move on to your second presentation, entitled Bacterial Community Response to Forest Clear Cutting. Please.
Hello again. This time I will talk about bacterial response to forest clear cutting, which is a part of my PhD that I'm currently doing at the Institute of Microbiology in Prague under supervision of Peter Baldrian. Before going into the bacterial community composition, I would like to point out that this study or this experiment started some time ago in our group, and it was already shown that the patterns of fungal community composition, biomass, also enzyme activities after the clear cutting of Pitta abies stand were rather complex. So the soil was sampled for the total of 2 years after the clear cutting, where bulk soil, rhizosphere, and roots were analyzed separately. Now, the main previous findings in this study were the decline of the mass of the fine roots. It decreased by almost half 2 years after the clear cutting, and together with the increase in fungal biomass during the peak of the root decomposition, this pointed to the importance of the roots as a carbon source for saprotrophs. Moreover, there was an increase in enzyme activity of many enzymes in the roots in the early stages of decomposition. So all of this showed us that the root litter represents a new decomposition hotspot for fungi. So naturally, we wonder what's happened with bacteria. What about bacterial communities? And here we can see on this NMDS graph that there is a distinct bacterial community on the roots. We're shown here in triangles with more profound changes in time, showed with different color. compared to the soil and rhizosphere here. And if we look over to the relative abundance graph of the most abundant taxa, we can almost immediately notice that the roots seem to harbor a more dynamic bacterial community. And this can be confirmed by looking at the specific taxa separately, like Penibacillus, for example, or Burkholderia, and their average relative abundance, which was higher in the roots shown in blue here compared to rhizosphere and soil. And this points to their importance and possible involvement in the decomposition of root litter alongside fungi. And now looking at the indicator species analysis here on the example of roots. So we are looking at an NMDS graph where these ellipses represent 95% confidence intervals, and the red colors represent early stages of root decomposition or the early months of the sampling, while the blues represent the later stages. The size of the gauges correspond to the relative abundance of the taxa. So we can see that many taxa are indicative of the later stages of root decomposition. For example, Bacillus among the most abundant ones, a common root colonizer, was indicative of the latest month, while Dechloromonas, commonly found in soil, was indicative of the first decomposition stages. Together, our results indicate that clear cutting indeed leads to profound changes in bacterial community composition in the roots, and that specific bacterial taxa are potentially involved in the decomposition of the roots alongside fungi, and they are possibly interacting with the fungi. The change in time seems more dynamic in the roots compared to the soil, complementing the previous findings of our group. With that, I would like to thank you again for your attention, and I will be ready to take any questions. Thank you.
I think we can't get Tiana leaving us without a question answered. Actually asking her another question, so I will do it, obviously. Do you have any indication of the role, or at least the presence of so-called endophytic bacteria, endophytic fungi within your communities, either bacterial or fungal? So what would you say? Do endophytic bacteria and fungi really play a significant and obvious role, or they are just a kind of small players in the whole community structure or decomposition, as in previous case?
Thank you, Tina, for the question. I'm always happy with any questions. I can say for sure about endophytic fungi, as this was already shown in this published work by my colleagues. And it was shown that indeed endophytic fungi have a role in these early stages of root decomposition as their relative abundance increased in the first months following the clear cutting. However, I cannot, at this moment, I cannot comment on the endophytic bacteria as this is something I yet have to look deeply into and see. It is a little bit more tricky for bacteria to assign ecology to the genus level. So this requires a little bit more expertise, most likely from my supervisor, which will come in the following months. So I will be sure to update you on that.
Okay, Tiana, thank you very much.
Thank you.
We will move on to the next speaker. The next speaker is Erna Vastak. Her presentation is entitled Assessing the Negative Effects of Severe Powdery Mildew Infection on Chlorophyll 2A Fluorescence and Stomata Characteristics of Quercus robur. Please.
Hello, my name is Erna Vastak, and I am a junior researcher at the Faculty of Agriculture, University of Novi Sad. I will tell you a short review of my presentation entitled Assessing the Negative Effects of Severe Powdery Mildew Infection on Chlorophyll A Fluorescence and Stomatal Characteristics of Quercus robur. Namely, the aim of the present study was to investigate the effectiveness of chlorophyll A fluorescence transient and chlorophyll A fluorescence kinetics in combination with stomatal characteristics for evaluation and monitoring of the severe biotic stress levels caused by the powdery mildew fungi. For this purpose, a semi-controlled greenhouse experiment was set up and constituted of 10 one-year-old disease-free seedlings of Quercus robur and the same number of specimens with 50 and 75% of leaf coverage by epiphytic mycelia. During the experiment, the infection progressed by its natural course, and at the end of the experiment, the first infection treatment group has reached to 75, while the second has progressed to 100% of leaf coverage by epiphytic mycelia. The results show that all parameters of chlorophyll A fluorescence transient were significantly negatively affected by this obligate leaf pathogen. Namely, all parameters of the OGIP curves were also affected. Furthermore, the performance index suggested the loss of density of reaction centers. In terms of slow fluorescence kinetics, the reduction of non-photochemical quenching indicated the disturbances and damages of photosynthetic apparatus, which resulted in incapability to transport the absorbed light energy to PS1 as non-photochemical quenching. Furthermore, we have noticed that the impact of biotic stress on chlorophyll A fluorescence parameters depended also on the intensity of infections. In terms of stomatal traits, the decline of stomatal GAR cell width in combination with increased stomatal density was pointed out as a protective mechanism of Quercus robur against this stressor. And in the conclusion, we have concluded that chlorophyll A fluorescence techniques are reliable, non-invasive, and non-destructive methods for understanding the responses of photosynthetic apparatus under severe biotic stress levels. Thank you very much for your attention.
Thank you very much, Erna, for the presentation. I think we'll have to move on to another poster presentation by Ilaria Fracasso. I'm sorry. Title is Turning Well-Known Climatic Archives into Biological Archives, an environmental meta-barcoding approach. Please.
Good afternoon, I'm Ilaria Fracasso, and today I want to show you a possible solution to turn well-known climatic archives into biological archives. As everyone knows, global warming is threatening our ecosystems, and it is affecting living organisms by reducing the genetic diversity, by changing community composition and the species equilibrium, and by causing local extinctions. For this reason, nowadays it's very, very important to identify the link between climatic, environmental, and community changes in order to understand the future of our ecosystems. To achieve this goal, we tried to turn peatlands into biological archives using the environmental DNA metabarcoding approach. We selected peatlands because they have been widely used in the past as climatic archives, and because they are able to store metals, particles, pollutants, and also DNA for 100,000 years. Moreover, from this stratigraphy, it's possible to infer a precise time scale. First of all, we collected a 1 meter long peatland core from the Lupicino peatland near Bolzano that has been divided into different slices. And for each slice, we performed the radiocarbon dating and environmental DNA extraction. DNA have been amplified and sequenced, and the results have been analyzed using QIIME 2. As metabarcodes, we selected the tRNL and the CO1 to identify plants and invertebrates respectively. So what we found? From the radiocarbon dating, we found that our core encompassed the last 1,500 years, but we also found that around the depth of 50 centimeters, there is a hiatus that caused a shift between 300 years ago and 700 years ago. We don't know why there is this hiatus, but we think that it could be linked to the use of peat in the past to produce heat. Regarding the metabarcoding, we were able to identify 18 plant taxa with the tRNL, but only 7 at the species level. While with the CO1, we were able to identify 23 different taxa, but only 11 at the species level. So to conclude, peatlands have proven to be available sources of environmental DNA, and they can be used to reconstruct these historical changes. Moreover, the eDNA metabarcoding can reduce the time of the analysis and does not require an high taxonomic expertise. But apart from these positive aspects, we also found some negative aspects. For example, we found a preferential amplification of some taxa, for example, Sphagnum for the tRNL and Rotifera for the CO1. And also this approach presents a low resolution in terms of taxonomic attenuation. So what we want to do in the future is to design some blocking primers to avoid these preferential amplifications. And we want to use different primer pairs and reference local databases in order to increase the resolution of our classifications. Thanks for your attention.
Thank you, Ilaria, for your presentation. I would have a brief question for you. What is the time scale that you can basically divide between two different points in terms of sampling eDNA? So how densely can you sample with the DNA in time-wisely manner?
Sorry, can you repeat the question, please?
Okay, yes, I will try to simplify it somehow. So when you're sampling your samples for eDNA,
yes,
you go deeper and deeper, I guess. And what is kind of the nearest time period that you can catch with two different samples? Do you have any experience?
Okay, it's not so easy to answer this question because during the years, I don't know if I'm going to take 3 centimeters in the deepest part of the core. These 3 centimeters encompass hundreds of years, but in the near the surface, the same 3 centimeters could be related to only 1 year. So the time for every subsample can change.
Thank you very much. I would ask now Luca Dunic to give us the last presentation of the early afternoon session. The title of his presentation is Isolation of Exogenous Fungal DNA from Non-Invasive Samples. Luca, please.
Hello to you all. My name is Luca Dunic, and I will present to you our study on the isolation of exogenous DNA from non-invasive samples. Mycorrhizal fungi are siblings of a vast majority of terrestrial plants. The symbiotic relationship allows the exchange of nutrients, along with other benefits of mycorrhization to the plants. Large mammals with their processes, like nutrition and excretion, are important vectors for the transfer and distribution of fungi between different environments. The aim of our study was to develop a method for isolating fungal DNA using commercial kits and amplify DNA by PCR. We isolated DNA from 16 roe deer and red deer fecal samples collected near the Lagatas-Velfinka highway section. We used 2 isolation kits to test for efficiency, the QIAGEN Stool Mini Kit and QIAGEN Parasoil Kit. Efficiency was estimated by measuring the concentration of isolated DNA and by the identification success using NS and albuscular mycorrhizal specific primers AM1 for the 18S region of the ribosomal RNA. From the 60 samples, 4 showed better amplification success. In terms of better PCR amplification, our results show that the soil kit is performing better than the stool mini kit. There was no significant difference in DNA concentration. By analyzing the length of amplicons, we revealed that feces of wild ungulates can be a source of fungal DNA. Future research should examine the amplified products using NGS, where we would expect most of our samples to amplify. This will allow us to study the potential animal dispersals of mycorrhizal fungi and other ecological interactions, like nutrition preferences and consumption of fungi, reasons of wild boar routing, and transmission of fungi by zoophily. I would like to thank our funders and also thank you for your attention.
Luca, thank you very much for your presentation. If there is no questions, I would like to close this early afternoon session and invite you all to the coffee break. And we will continue at 5 minutes past 4. Dear participants of the Live Genmon conference, welcome back to the last session of this afternoon. The title of the fifth session is The Science Policy Interface. We will have 3 keynote speakers, 1 oral presentation, and 4 poster presentations. Now I would like to invite Michele Bozzano to join us live and present us his talk with the title Genetic Aspects in Production and Use of Forest Reproductive Material, Coordination and Dialogue.
Hello, can you hear me?
Yes, we can hear you. Please switch on your webcam as well so that we will see you too.
Yes.
Perfect. So you are on.
Okay, thank you very much.
I would kindly invite you to share your presentation and give us the half an hour lecture. Thank you.
Thank you very much. So first of all, I would like to say that I was somehow pleased to learn that I had to give this, this presentation live after the original file of the recording was corrupted, because I find it much more engaging to be able to have people online directly listening to what I'm bringing and I'm sharing here. Let me see if I can share my screen now. I hope you can see the PowerPoint, not the notes, but the full PowerPoint. Some nodding will help. Yes.
Yes.
Okay. Thank you very much. Thank you very much for the Live Genmon project for inviting the UFOGEN program to this to be present this event. We are very honored to have this possibility and also to be able to follow up this very interesting and engaging conference, which really brings together many scientists, and also it really makes a summary of genetic monitoring within the forestry domain in the past 5 years. Before I continue with my presentation, I would like to give a very brief outline of what the UFOGEN program is. So UFOGEN is a collaborative program on forest genetic resources, which is financed by its member country. And you can see in this map here the current membership. It ran on 5-year phases, and we just entered the 5th phase of the program. UFOGEN developed strategy, guidelines, and recommendation for the conservation and sustainable use of forest genetic resources. We have been producing various kinds of products through the past 25 years. And what I will present today is one of the most recent publications that we are just about to release on the genetics aspects in the production and use of forest productive material. What we've been doing within UFOGEN, it was to collect the scientific evidence, to collect all the scientific knowledge that we have in this field, and we try to prepare it in a way that is available for the development of decision support tools. But before I continue into details, I would like just to spend one slide to say why I think that coordination and dialogue are really crucial in the field of genetic resources, and both the conservation, especially the forest productive material production and sustainable use. So first of all, we face a continuous evolution in knowledge and in tools that we are available, especially for the practitioners. So these tools are really important that they are developed in a way they really meet the need of the practitioner on one side, and on the other side that they're really very quickly disseminated. We have a climate that is changing very quickly. There are actions that need to be taken by practitioners and need to be ready to face things that were not prepared during the academic or the technical career. So we really need to have a very close communication, a very close engagement with the practitioner. We need coordination because the framework, the political, the regulatory framework that rule and then overseeing all the various aspects from the management of the forest to the artificial regeneration or whatever, it needs to be really adapted to the current situation, adapted to the new challenge, supporting the new knowledge, and especially supporting the practitioners. This because the decision, they have to be taken at local scale. We have forest managers that are in the forest that they need to decide which species to favor, what kind of treatment apply to a forest, what kind of material to use in artificial regeneration. And all these decisions have taken locally, but they need to follow the bigger picture. You know, the local practitioner cannot see how a species is moving simply looking at the local scale, cannot see what is the bigger framework, but they need to be supported by this global vision. And to do so, I will just like to give some very small national or sub-regional example, just 3 slides simply to bring you through some, some thought about this field. The first one, it comes from the SUSTRI project that Silvio Schuller presented on the first day of this conference. Within the framework of this project, what they've been doing, they've been observing species, their current distribution, and how the distribution range of species was going to change. So they modeled the distribution and seen how both geographically and ecologically the species distribution was gonna is foreseen to change in the next years. Based on this, they've been developing some tools. In the case of SUSTRI, was really an app that was available for the practitioner to select what was the most appropriate material to be used in the virus site. So basically, this kind of tool really empower the practitioner in having choice, in seeing, in understanding the bigger picture, even if they have to take decision at the local scale. Another nice example comes from France. In France, they have decided in the moment they were developing the seed zone for the various species, not to have a standardized seed zone, but to have a seed zone for each species differently depending on the genetic of the species, on the ecology, and its actual distribution. Up to 2015, if a plantation was supposed to be done in this big seed zone here, highlighted this key PE12112, the traditional, I mean, the existing guideline until 2015 was to use either material from this seed zone or from this neighborhooding seed zone, and that was it. In the moment, they realized that there is a climate happening. There is the scientific knowledge will show that Quercus petrea is supposed to shift from south to north. Then the guideline for the practitioner will also foreseen that there are other seed zones that could be used in the highlighted seed zone. So this is a tool that is made available for the practitioner in understanding, okay, this is what's happening. These are the seed zones where I can look at. Next example is just, a very small experience that I've been going through this summer. The dot is a site where I wanted to plant some Laris decidua, and then the triangle is the nursery that it was a regional nursery that I've been approaching in order to get the reproductive material. Now you see highlighted the distribution range of Laris decidua, and then here you can see in the dots, you can see where are all the seed zones that actually come from the Evogene information system that we are managing. Now, my expectation was that nursery for the site I was asking for, they would provide material from the seed stand that is very, very close by. But you know, this material, when it's commercialized, then it has a documentation. So through documentation, I could read the code, and from the code, I could come back to the origin of the material. And to my surprise, was from the other side of the Alps, and actually coming from a forest that was growing above 2,500 meters, planted in an area that was completely not matching the ecological niche, and especially not taking into consideration any kind of assisted migration of the species. So this is a real missed opportunity, and especially this is also show why coordination is really needed. This material should not have never been made available in that nursery to be planted in this part of the Alps. This is just one example why to simply highlight the fact that a coordination, a global vision is needed. We cannot just let the individual actor to work on whatever is available or taking decision without seeing the bigger picture. Now we come back to this publication. This publication is being finalized those days, and I will say that within the next month is going to be available on the Evogene website. Again, our effort was in going through all the literature, all the knowledge that we have available. We went through a number of workshops, a number of coordination between the various actors within the community in order to highlight and identify what are the various aspects in the production and sustainable use of forest productive material. What I will show now are few of the recommendations that I think that are relevant for this audience. So the first one that we know that especially in some European country make people jumping on the chair is that natural regeneration is not always the best option. This, I don't say that is the best option, I simply say that is not the best option. And this is something, is a concept that is really important, we present, that it is an alternative that we need to make available for practitioners and owners, so they are aware that they can have this alternative, but it's something that's really important and they need to have this alternative. And this is something really important we need to communicate for other actors, like some of the main certification schemes that we have currently in Europe, that very often they do not allow to have artificial regeneration for more than 1/10 of the total surface of the stand. So this means that forest owners, they don't really have this option. Forest owners, they just have to go with natural regeneration. Okay, sometimes, as I was saying, natural regeneration normally is the best option, is what we advocate for, because of the huge amount of seedling that are going to be planted, I mean, naturally planted there, and through a selection process, they will generate a forest that is really adapted. But sometimes it's not possible, either because there is competition with other species, while we want to favor one species against the other, or simply because some provenance of the species, like we were seeing later on, should be better adapted to the site. So it's really important that this option is available. Having this option available, promoting artificial regeneration as one of the options will give a side effect, which is really important, because it will support a viable nursery system. We have seen in Europe, we've seen in Slovenia, we've seen in Italy, in France, Sweden, many other countries, that we can have natural disaster happening from one day to the other and having entire forest completely wiped out. In those moments, we need to have a nursery system in place. We cannot just make it up from one day to the other. And we are in the moment we realize that artificial regeneration is needed and nursery system is needed into the forestry system, then allowing the two, I mean, supporting both things happening together is really a very nice backup plan, is a very important element in having a healthy forest in Europe. Another recommendation will be on the appropriate choice of forest productive material, because forest productive material need to be genetically fitted to a specific site. And this is really important. We see what kind of forest we have. We see what kind of material is naturally going there. And having an option to look at other origins of the reproductive material and being able to match the ecological niche of where the forest, the current, the place where we want to plant it, and the origin of the productive material, it really help us in matching, in planting something that has a very high likelihood to be able to survive in the long term. Another important element about the reproductive material is that it needs to be sufficiently diverse from a genetic point of view. And this to allow the new forest to have the capacity to adapt to a changing environmental condition. Until 2 years ago, very often the forest managers, they were looking for very narrow genetic diversity in order to have a production at the end of the term that was simply having a very uniform stand to be selling in the market. Now what we have to look at is to be sure that something will really survive. We need to differentiate our portfolio. If I can make a comparison, if we think about forestry like our saving in the stock market, we can think that we can either decide to put everything in something that it can give us a very, very high gain, or try to differentiate our portfolio. We may not get this very high gain, but we are sure that we'll get something at the end of the cycle. And in the moment that the market is very unstable, then this is the best option that we can give to to the investors. With using this comparison, we can say that for a forest owner, for the forest management can advise, don't use something that it has a very narrow genetic base, use something that is very flexible. But this is not the one rule. This is just important. It is one of the rules that can be used, because in many other cases, especially when we have very precise knowledge, we have very good forecast and very constant climate or even short rotation cycle, is still very, very good and very fine, narrowing the genetic base and have something that can be managed in one way. It's just, again, it's just about having many options. And as I will say in the conclusion, what is really important is to have capable practitioner. But now we come back to another recommendation that very often raise a lot of concern in the audience. The point that local is not always the best. Okay, we know that the autochthonous local tree population originate for a very complex selection process, and but local population, they don't always demonstrate to have the optimum fitness compared to other reproductive material. So for this reason, since local environmental condition can change, forest managers should extend the option to non-local reproductive material. And this very well linked to the next recommendation concerning the use of provenance instead of, changing provenance instead of species in the system migration schemes. This one I think I need to clarify. We know that adaptive genetic diversity within forestry species is always very, very high, or it can be very high depending on the species, but sometimes it's really very high. What's happened, what we've been observing is that very often forest managers, when they see that a species in a stand is not performing well, they try to simply to replace the species with another species. Sometimes it works, there's nothing bad with that, but what is important is that among the knowledge tool and among the practice that the forest manager need to have is the possibility of simply replacing that species with the very same species, but from a different provenance. This because the flexibility, the plasticity of the species in another part of the distribution range could very well fit the environmental condition that we have in the site. And this one, the important implication in this action is that the ecosystem will not be changed. The ecosystem will not be altered. It will be the very same ecosystem that will simply evolve in the same way, and the new provenance will simply go to take the very same niche of the original population. I'm about to summarize with a couple of slides. So what is needed? So the first thing that is needed is that we need to develop decision support tools for practitioners to support the conservation at all levels. So conservation of forest genetic resources need to be done at the level of the landscape. So each practitioner, in each landscape, there is some level of genetic diversity that is available for the natural regeneration, is available as a local seed source, is available for all the adaptation process of the local population, but is also needed at a very large scale so that we make sure that we maintain the adaptive potential of the species. Is important, those decision support tools are important to guide the selection of forest productive material. We really need to make sure that we keep developing tools so that the practitioners, they have tools in their hands so that they can choose the appropriate reproductive material. On the same way, we have to work with policymakers because they need to develop, they need to support a decision makings of the practitioner through a regulatory framework. It's really important that the practitioner, they have all the possible options. Again, on what is needed, we need to have a dynamic forest nursery sector. As I mentioned before, a forestry sector is very important for the artificial regeneration, but even more important for in the moment we have an emergency, when we have a catastrophe happening in our landscape. And we need to make sure that this system works potentially, but also in practice. We need to have a coordinated production and distribution change of forest productive material. This, as I was saying before, and this is one point that we are really strongly advocating. We need to see the bigger picture. We need to see how the material is moved. Ideally, we need to track how the material is moved and to keep record for very, very long term in order to be able to assess what was done and the success story as well as the failure. This kind of coordination, as I was saying before, when there are actions that are taken at a local level, but they need to reflect the vision that we have on what is happening in our landscape, what is happening in our region. And with this, I conclude with what I consider one of the most important recommendations. We need to have qualified practitioner. We need to find a way to support the practitioner, because are the one that taking action, are the one that they really can enable this change to make sure that the forest can produce timber, they can produce ecosystem services. We have forests that they are always very healthy and capable to adapt. But to do so, we need to create tools and we need to create alternative for them. So all the tools and alternative are not replacing a very well-trained practitioner. They are simply a tool in their hand so that they can really take the decision in order to support the forest, that they can deliver all the services. The services, again, from the production of very high quality timber and to provide ecosystem services. And with this, I thank you very much for this opportunity. And I hope the quality of the audio was decent, and I'm happy to reply to any questions. Thank you.
Thank you very much, Michele, for this presentation. We have got 2 questions in chat. So for the practical reason, I think I will read the first one and you can answer it directly. So you say that nurseries with the right material are needed in case disease happens, or disasters happen. Makes sense, of course. How can forest nurseries be stocked with the right material, given the way acquiring the material works in the European market economy and mostly on a short notice orders? The question was posed by Bruno Fadi.
Thank you very much, Bruno. Thank you for this question. Actually, in the first version of this presentation, I was also including a small simulation about the time frame that we need to operate. This is a big change we have to undertake. Most of the action that we do very often are on a very limited time frame, 2 or 3 years. We have to accept the fact that a nursery system cannot work with such a small time frame. From the identification of the seed source, if you want to have the right seed source available to be planted in a site, we need to think on a very long time scale. So it means that the nursery system needs to be able to operate on something that goes above the 10 years time scale. And this is something that is really important to have in place, that we can order material coming from needed to be planted in one site. It needs to be ordered, and then we need to be ready to cope with a very long, with a longer scale, not pretend the material is available the day after. If we know that we want to plant somewhere, we need to have both, again, a regulatory framework that allow the resources to be used in the long term, if it is a disaster or whatever, or if it is a simple regeneration, we need to train the practitioners so that they can pre-book many months, many years before the moment they can use, especially if they want to have a specific provenance. In the moment the system is in place, then there are many more provenance that they are available. Of course, when a natural disaster is happening, we need to work with whatever is available, and we cannot pretend that the nursery system is available from one day to the other to deliver whatever we want. We need to accept the fact that there is a gap between the moment we realize that we need something and this material is available.
Okay, the second question was asked by Robert Ekblom. He asked if using only data on place of origin as a proxy for the genetic profile of the stock would be a good option, or you would prefer to use direct genetic data such as genotyping and sequencing.
Unfortunately, for most of the European tree species, we have very limited genetic knowledge. So in some cases, we can really go to very precise recommendation, but this is limited to a handful of species. In the moment we want to be prepared for, we want our forest to be prepared to face climate change. We need to have to increase drastically the portfolio of species that we have in the landscape. So in this case, in most cases, what we can have is simply an ecological characterization. Of course, in the moment you can be very precise, we can identify what are the traits that we need, then this goes again in the direction that we really need to have a close gap between science and practitioner. In the moment a new knowledge is available, in the moment new characteristics of species are being revealed, then those need to be immediately changed into support tools for the practitioner, so that we can support the practitioner in taking their own decision based on the new tools that are available.
Okay, thank you. I see no more raised hands, but actually I have a kind of challenging question for you, maybe.
Please go ahead.
The Elforgen is now working with forestry species, more or less the more important or the dominant ones. Do you see any future in using the same principle also for, let's say, non-timber forest products, such as healthy plants for food like berries or maybe mushrooms?
U4Gen in this moment is working well. Theoretically, any species growing in Europe is any forest, any timber forestry species is relevant. In this moment, we are working on about 110 tree species, which are from the most relevant one, the most ecologically important one, and economically important one to the less important one. And this is the focus. This is where we have the mandate. We do not have any mandate on medicinal plants, on berry trees or fungi. There are other communities that have this mandate, that have this responsibility. What I have to say is that we are working in close collaboration with different actors involved in conservation of different kind of genetic diversity. This is because we found more and more that is really important, like it was saying actually in a presentation in this conference earlier this week, is really important to conserve the entire ecosystem. Having an entire ecosystem, an entire healthy ecosystem is a very good way to maintain high level of genetic diversity. Is also a very good way to maintain the genetic diversity of a target tree species. But no, U4Gen is not planning to enlarge the mandate to other outside the forestry species.
Okay, thank you very much. If there is no further question, I would like to thank again to Michele Bozzano for his presentation. And we would continue with the second keynote speaker of this afternoon. She is Eva Hermanovic. The title of her talk is Storytelling for Science Communication and Media Engagement. Eva, please.
Hello, good morning, everyone. I'm Eva Hermanovic from the European Forest Institute, more specifically U4Gen program, where I lead the communication activities. And also I'm responsible for communications in other projects related to genetic resources. So thank you very much for inviting me here. I'm very pleased to be here. I'm sorry that I cannot be with you in person and engage with you in a conversation after this presentation and also hear directly your questions. However, this is the circumstances that we are living right now, so we are making the best of the technology that we have at hand. So thank you very much again for inviting me here. And today I would like to talk to you about storytelling for science communication and media engagement. And I will give you a couple of examples of how we effectively use this approach in communication plans of the U4Gen program. And I will also talk to you a little bit about breaking down the concept of storytelling. So just to start, everyone knows what a story is, and everyone has listened to many stories in their lives. And we all know that storytelling has the power to engage, influence, teach, and inspire listeners. That's why organizations should build a storytelling culture. There is an art to telling a good story, and we all know a good story when we hear one. But there is also a science behind the art of storytelling. And I would like to start here with some science. These are formulas, and I wonder, this is a little quiz here for you. What could these be? I can give you a hint, we are in the field of neurochemistry, and this is somehow related to storytelling. So I cannot hear your responses. However, here I will reveal what this actually is. So the first one is cortisol, the second oxytocin, and dopamine. These are three hormones that scientists are discovering that are released in the brain when we are told a story. And why does this matter? If we are trying to make a point stick, cortisol assists with formulating our memories. We very often don't remember series of facts, but we remember stories, and this is how we remember the facts that were told in the stories. When it comes to creating deeper connections with others, oxytocin is associated with empathy, an important element in building and deepening or maintaining good relationships. This is what is created when we actually empathize with a protagonist in a story. We relate to them and the oxytocin is released. Dopamine helps us regulate our emotional responses and keeps us engaged. So this is why, for example, when we watch a good Netflix series, we want to watch the next episode. This is dopamine steering our emotions. So staying a little bit again in the field of psychology, to understand how our brain works, there is this nice metaphor by Jonathan Haidt from the New York University, and it's the metaphor of an elephant and the rider, where the elephant is our emotional brain and the rider is our rational brain. So very often we think that we take our decisions completely rationally, but we have to remember that the big steering beast that is driving us on our path is this elephant. So we do have some control, and the secret here is to find a balance between the rider and the elephant. But we always have to remember that the emotional part of us is very strong and very, very big. So there is also very often a situation where it's our emotions that are taking us wherever they decide. So what is actually a story? In a nutshell, it could be summarized that it's something like that has a beginning, a middle, and the end. It can be illustrated through this narrative arc where the chronology of facts takes us from the exposition through some kind of climax towards denouement, or where things get sorted out and resolved at the end. It can be also shown differently as a story structure. So in the beginning, we have the protagonist with whom we empathize. Then in the middle, the protagonist is encountering some kind of challenge and a struggle. They might be living through some kind of personal crisis as well. And in the end of the story, there is some kind of climax, a revelation and a resolution. The narrative arc and the series of facts, the protagonists are not everything in a good story. There are also some building blocks which appear very often in a good story. They don't have to be there all at the same time. However, a good mix of some of them makes the story much stronger and much more powerful. So there is drama, there is suspense, surprise. There are some universal values that people can relate to, high stakes. So the story and the challenge actually matters. And The character has some needs and some flaws. There is action because we don't like boring stories. There is some detail, and there is also authenticity, which is very important in our science organizations, so that we make sure we don't tell stories that don't sound authentic, that sound like scripted, but rather there is an authentic element to them. And what does the audience get out of that? The feeling of catharsis, which is basically a release of the emotions and the tensions that are created from the beginning of the story, where we see the challenges rising and then the struggle somehow coming to an end, in a bad or a good way. So a few years ago, we produced a short film for the U4Gen program, and this is the first example that I would like to give you for telling how we can actually use storytelling approaches in our everyday communication, science communication work. And this film was picked up by the National Geographic, and this has actually led to the film going viral on the website. Here are a few statistics just to show you the reach of what it brought. So there were over 3.5 million views, a lot of comments, which were actually proving the fact that the story was a very important element as to why the audience engaged with it so much. So this is one of the comments of the over 4,000 comments that I picked, is saying, this is so touching. For the first time, I've been touched by a tree story. So people really do see the value of the story, and even those who are not specifically so interested in forest genetics could somehow relate to it and see the elements that were relevant for them. The publication led to a lot of press coverage that got interested and covered the story in their various media and articles. I just selected a few press clippings here for you to see, but there was a lot more than that. And there was an article that went together with the short film that was published, and it was reused very often in the various coverages by different media. In terms of impact, there is also a few highlights here that I wanted to share with you. There was certainly increased funding from the government. Foreign funders also got interested in afforestation, and just citizens around the world who were reaching out and wanted really to get involved in some action. It was really leading them to want to engage with the afforestation. And the main character also appeared in a 10-part series on TV on global warming, and had another proposal to be part of a BBC Nature series on plants. So himself also was a very successful character, and could then go on and engage with other media and talk about his topic. So the second example that I wanted to share with you is another film that we produced and published earlier this year. I will just show it here with you. We haven't really had occasions, due to the given circumstances, to screen the film at various events. So it's too premature to to share with you any statistics and the impact that it might have had, because we are still in the phase of releasing it and showing it around. And the audience here today is one of the first ones to see it in public, even though you were watching it from your own computers. As we watch it, I would also like you to think about the elements that I mentioned before on how the story is built and the different building blocks that make for a good story. And of course, I invite you to just enjoy it for yourself, but also in the back of your mind, think about how the different building blocks were applied in the production, so that you can see how this is all working in practice. So I will now play the film. It's just 3 minutes.
20 million years ago, there was a huge volcanic eruption in the part that today is the Lesbos Island. Lots of lava started to flow, ash that covered the subtropical forest that existed at the time, started the fossilization process. And by that we know that we have an ancestor of today's black pine that was living here at the time, Pinoxylon paradoxum. I was born in Lesbos. I was raised on this island. When I first went up in the mountains, there were two big black pine populations. One of these populations is almost gone. We may lose them. The second population, which is still in very, very good condition, which has to be preserved, is the speed of environmental change that scares me. With the climate change, you have fires occurring in the mountains where black pine forests grow, and a very hard time for this species to adapt against its competitor, another pine species that grows in lower elevation and is coming up. Black pine doesn't have enough time to adapt in this kind of situation. Losing them is not only losing a few trees, is losing all this genetic heritage going on for all these millions of years, not only on Lesbos or in Greece, but all over Europe and the greater Mediterranean area. The local forest service, they're engaged in forest protection activities, fire prevention activities, removal of ground vegetation. Under very, very limited means, they do whatever they can to facilitate the survival of black pine forest on this island. The broader the genetic base, the larger the chances of a species being adapted to some sort of steady state. It's important to remember that this is the birthplace of Theophrastus, the birthplace of botany. Aristotle came here as well, and together they studied the fauna and the flora of this island. So it's very important that we continue this legacy and we try to study in depth these species and maintain their populations for future generations.
So that was the short film that we produced to show the importance of genetic heritage and the forest management to maintain the genetic diversity. I would like to also take this opportunity to thank Phil Aragonopoulos for his participation and active engagement in the production. Both films are available on the U4Gen website and the U4Gen channel, so you can easily go back to them and analyze them for yourself with the elements of the storytelling. I will be very happy to hear any comments and respond to any questions that you may have related both to my presentation, but also in general to storytelling and how to apply it in the communication strategies of your organizations, especially related to science communication. So thank you very much, and I would like to welcome any questions that you might have, both here via email, via Twitter, and in the Q&A session after this presentation. Thank you very much.
Evie, thank you very much for your presentation. Katya asked me to read her question now. She said, do you think that live projects could reach a critical mass needed for change with a storytelling approach?
Yes, of course. Why not? Every project could reach a critical mass if you find a good story and a good character. I think it doesn't matter what the topic is, but the important thing is to find out the story, which is the most difficult part, because it doesn't only have to be a story. Of course, it has to be very strongly linked to the key messages of the project. So that's where the most difficult part lies. And I see that there is also a question here from Bruno saying, how do you suggest we build empathy for the barbaric term FGR? I think this is the area that I have worked most, and I hope to have built some empathy. So the response would be through, well, the empathy is usually something that we feel for people. So it has to be through the person who is the protagonist that we build this empathy. And we have to just remember that they should not only be knowledgeable about the topic, but also be good communicators that people will want to listen to.
Perfect. It's obviously clear that scientists, we need someone who would help us telling the stories, because at least from my experience, I think we are quite robust from time to time in putting out our ideas and our achievements.
Yeah, but it's also up to the communicators to do a good interview and to take the good part of people, and then to decide how to do the edit in a way to take just the parts that are resonating with the public most. So it's also the communicator's job, and a lot of things that happen behind the scenes when producing this.
Well, Evie, thank you very much, and hopefully for lots of successful stories. Our next story will be told by Katja Sonnenschein about the LiveGen1 communication and dissemination activities. Katja, please.
Hello, I'm Katja Sonnenschein from the Slovenian Forestry Institute, and our responsibilities are communication and dissemination activities of the LiveGen1 project. In this presentation, I would like to communicate to you how we have communicated the importance of forest genetic diversity and forest genetic monitoring to different publics, to general public, and to special target groups. To general public, we have been basically explaining the picture that Domen Finchgard drew while working on the LiveGen1 project.
that the ecosystems rely on biodiversity, which starts at the lowest level, the G. The genetically diverse forests have better chance of surviving in times of climate change. That's very general. And to the special target groups, for the others, different approaches were taken. We communicated to forestry professionals, foresters, students, and policymakers. We didn't focus on explaining the pyramid, but rather on the process of forest genetic monitoring and the benefits of it. I will first explain the approaches that we have used for the first group, for the general public. We have even managed to explain forest genetic diversity to preschool children, but let's first focus on grown-ups. So we established the LiveGen webpage. Here are 5 boxes which you can visit. This one we will see later. Here you have the news, the portal, which brings you to this one, materials and events. So if you visit the livegerman.si, you can learn about the projects, you can see all the materials, and you also have all the links to the social media, which we mainly use Twitter and Facebook. And as I said, it takes you also to this portal where we gathered some knowledge about forests. There are 4 boxes: learn about forests, teach in the forest box for forestry professionals and students, and forest owners. So if you click in these titles, you will receive material from which you can learn about forests, about the importance of forest genetic monitoring. And then we also established the conference webpage, which you have visited before. The information is not available only on the internet, though. Among more visible printed publications are children books and handbook for learning and play. These I would like to show you. I'd like to show you where you can find them and download them. Let's see. Sure. Here are the cartoons that tell you about the books. And here are the books in English, Slovenian, Greek, and German. We can take a look at one. See, you have the book, and here you can find the Big Forest app. This one makes the book even more interesting. It makes it alive. The trees pop out, the birds start singing. So this is one app that we developed. And later I will explain you about the other app that we developed in this project. If we go back, we will see also the other very successful book, which is a handbook for learning and play in the forest. It's a teaching material. From this handbook, you can learn about especially about the games that you can play in the forest and learn from them. They're meant for teachers, but also parents that would like to teach and entertain children and lure them to forest so they would enjoy it better. If we go back to the presentation, let me see. Okay, you can see another app, mobile app that we've developed, the Seed Hunter. I will explain it now. It's the AVG developed it. It's a forest pedagogic game for a smartphone. It's for the Android, and it goes. The Mark Walter will present it today later. So in more detail. So if you're interested, please listen to him later. So the main concept goes like this. The player goes on the hunt for tree seeds. The seeds are over 3,000 virtual objects, which are placed inside forests and parks. And as soon as a player approaches a virtual seed, she can collect it with a smartphone. An international ranking list shows the user's success, so you can compete with the others. What are the benefits, we think? The game takes place outside in forests and parks, and it has some educational value. It raises the awareness of the rare tree species and the tree species that are threatened with extinction. It has this collecting seeds in reality has an important task to protect genetic diversity, which we learn in the virtual space. And it gives us link between technology and nature, from smartphone to maybe truly growing a real tree later. Here we continue the description of activities for the first group, the general public. Today, Usha Wilhar will have a poster presentation of live GenMod educational activities. But I would like to only give you an idea that how successful we were in that part. Only with open days, for example, our project team reached more than 4,000 people. If we continue with the first group, we managed to publish more than 130 media pieces. Media reported on our activities in all project countries and also in Bosnia and Herzegovina, in Croatia, and in South Africa. And here we come to one of the more notable large project for general public, the film about the forest and climate change. It emphasizes the importance of genetic diversity to preserve the adaptive potential of forests to the changing environment. In addition, we have bought the distribution rights for the documentary Nature Parks of Slovenia, the Virgin Forest Krka. And I suggest that we take a look, a few minutes at both, and you can also see where you can find them. So if I go and share the screen again, I will go to the featured films. Here you can find them. Among featured films, you can find some videos, and then this movie in English, and the movie about Krka. So let's have a little sneak peek in the film Forest and Climate Change.
Iz aktivnosti Evropskega programa za gozdne genske vire EUFORGEN izhaja projekt Live GenMon. V Sloveniji, Nemčiji in Grčiji se izvaja od leta 2014. Namen projekta je razviti sistem spremljanja oziroma monitoringa genetske pestrosti gozdnega drevja. Med cilji projekta je tudi ohranjanje genetske pestrosti na območjih med Bavarsko in Grčijo.
Poleg testiranja in razvoja verifikatorja, se pravi meritev genetske pestrosti za posamezne drevesne vrste, kar obsega delo na terenu in delo v laboratoriju, pripravljamo tudi smernice za izvedbo monitoringa genetske pestrosti. Zato organiziramo tudi delavnice za gozdarje in predlagamo tudi spremembe in dopolnitve zakonodaje in strategij v posameznih državah, ki bodo omogočale izvesti genetski monitoring na različnih nivojih zahtevnosti v Sloveniji in v drugih državah, ki so za slovensko gozdarstvo zanimive tudi zaradi morebitnega povečevanja genetske pestrosti in testiranja gozdnega reprodukcijskega materiala iz držav jugovzhodne Evrope na našem ozemlju. Med najbolj konkretne ukrepe genetskega varstva gozdov sodi izbor ustreznih semenskih objektov, v katerih se bo pridobivalo seme ali puljenke, načrtovanje gospodarjanja s temi sestoji, načrtovanje pridobivanja gozdnega reprodukcijskega materijala, torej predvsem semena, ki se ga naj bi pridobivalo v času masovnega obroda.
Za genetske analize najrajši uporabljamo popke, ker je v njih največ DNK-ja. Zdaj, če pridemo sredi poletja, bomo najlažje vzorčili liste. V primeru pa, da je drevo zelo visoko in da listov v nadventivnih pogankih ne dosežemo, potem pa lahko vzorčimo tudi del skorje. Za genetske analize potrebujemo približno toliko izhodnega materijala, pa ne samo za analize, ampak tudi zato, da en del tega materijala shranimo za morebitno kasnejšo uporabo, posebej v primeru genetskega monitoringa.
I will now show you a few minutes of the other movie about the virgin forest.
The southeast edge of the Alps in Slovenia. Our forest has been able to survive here during the periods of extensive glaciations. A tiny slice of it is still preserved to this day, the Krokar Primeval Forest Reserve. Foresters entered this ancient forest just over a century ago. At the edge of the then almost untouched forest in Kočevska, at the ridge of Mount Boroška Gora, they stumbled upon a primeval forest, which has remained as it was back then, a primeval beech forest. This forest and the woods in its vicinity between the Alps and the Dinaric Alps are a gene pool of the beech trees, which have, after the end of the ice ages, inhabited Central, Eastern and Northern Europe. Amongst all known European populations of beech trees that have survived the glaciations, this population was the first to start spreading across Europe. Their seeds continue to travel even to this day. The ice cover of Europe has descended across the Slovene Alps to their mountainous fringes. The European beech has surely survived at least the last two most intense glaciation periods in higher, more sheltered areas in the so-called refugia.
With this slide, we leave the activities that target general public, and we go to communication and dissemination activities for special targeted groups. Forestry professionals, foresters, students, policymakers. Their understanding of forest genetic diversity importance, transfer of knowledge about forest genetic resources and monitoring was in focus of target communication and dissemination. We had workshops, meetings, we participated at numerous events. These efforts contributed to outcomes that were presented by Boris and Antasha today. We organized internal workshops for forest owners, users of forest reproductive material, tree dealers, nurseries, seed dealers, silviculturists, policymakers. The workshops on forest seed collection and handling, on forest and tree seedlings and their handling, on tending of forest stands, and on inclusion of forest genetic monitoring into policy in practice, revealed the need for the protocols on the seed collection and tree seedlings. They were later published by LiveGenMon. Andrea Gregoric presented them today in her poster presentation. At these internal workshops and other 14 events for similar public, we reached 600 people. We also had 10 workshops for silviculture and forest genetic scientists. Last year and this year, before the corona crisis, a series of workshops were organized in the Transect area. These workshops were organized in Croatia, Bosnia and Herzegovina, Serbia, and Republic of Northern Macedonia for promotion of forest genetic monitoring. We also had workshops and summer schools for students. 700 students learned about LiveGen1. In Germany, 4 events were organized with 180 participants. In Greece, 100 students participated in 3 events. And in Slovenia, we reached 400 students with 14 events. We also had common workshops with complementary projects and programs. Our project members connected with other projects and programs on several occasions. LiveGen1 is in constant cooperation with Cost Actions, currently GBike, and live projects, Live Systemic, Live Links, Live Artemis, just to name a few. LiveGen1 project team members participated at more than 30 scientific, professional, legislative, governance events. We presented project aims, topics, progress, and findings as often as possible to a broad spectrum of audiences. Therefore, LiveGen1 was presented at European Commission, UFRO and FAO meetings, congresses and conferences, ministries. As a result of all these activities, scientific monographies, proceedings of scientific meetings, and professional articles were published. Here are just a few of them on the slides. An article for genetic monitoring, an overview of concepts and definitions, for example, provides an overview of forest genetic monitoring concepts and definitions, discusses their advantages and disadvantages, and provides a flowchart of the steps needed for the optimization and implementation of forest genetic monitoring. And an article, Interplay between forest management practices, genetic monitoring, other long-term monitoring systems in forests, discusses the possibilities to combine forest genetic monitoring with other existing long-term forest monitoring systems. And this is just an example, and there are really many. You can also see in which magazines we've published. Another important group of publications are guidelines for forest genetic monitoring. Darius and Gregor had a keynote presentation on Tuesday on guidelines for conducting forest genetic monitoring of the 7 free species that are covered by LiveGen1. In the guidelines, we used illustrations that were done by 7 scientific illustrators. There was an exhibition of this work at the Slovenian Forestry Institute. And with this beautiful picture, I'm slowly ending my presentation. This is what LiveGen1 has done to communicate the efforts that we need to make to help our forests to adapt to the changing environment and climate. And of course, we must put an effort also in climate change mitigation to help forests from the other angle as well. Thank you for your attention.
Katja, thank you very much for your presentation. We are a little bit ahead of time, so I open the floor for discussion. Otherwise, I will have to ask a nasty question. Well, Katja, actually one rather technical question. Most of the people that work in science have an idea how much time it takes for a scientific paper or maybe to make a scheme or something like this. But how long does it take to make the movie, the kind of movie that you have showed earlier in your presentation? Is it a 1 week, 1 month, a year?
You mean the one about the forest and climate change?
Yes, for example.
Yeah, it's actually about 1.5 year work if you work on it here and there, not just focus on a movie, because you have to catch the sun, the right, you have to have the right timing for everything. There's a scenario, you need a good cameraman. And yeah, all in all, I would say a year and a half.
Fantastic. And you have managed to produce 2 within the project. Very good. We have a question by Phil, please.
Hi, Katja. Nice presentation. Thank you. My question is, if we were to start now a big project like LiveGenMon, would you do something differently regarding dissemination than we did already?
Yeah, perhaps. I would focus more on, as Eva said, on storytelling, because we heard how good it is and how well you can reach the public. And yes, I would put more effort into video production, because we, by our experience, it has a good response. And I would maybe use less printing of the material and actually put more effort into Zoom meetings to discuss how to make a good story and disseminate it.
Great, thanks.
What good acting.
Thank you.
Katja, thank you very much. Phil, thank you very much. So we are looking forward for your next TikTok clip, Katja. Thanks again.
Thank you.
Our next presentation will be given by Urša Vilhar. It's another story, a successful story about the general public dissemination of the LiveGenMon project. Urša, please.
Dear ladies and gentlemen, my name is Urša Vilhar and I work at Slovenian Forest Institute. Today, I will have a short presentation about the general public dissemination of the LiveGenMon project, which is a contribution of a large number of project co-workers in Slovenia, Germany, and Greece. The main focus of the dissemination in the LiveGenMon project was to promote knowledge about forests, forestry, forest genetics, and raising awareness about the effects of climate change on forests. In order to achieve the most efficient transfer of knowledge, dissemination activities were divided into main target groups, taking into account differences in age, interests, and level of knowledge. The main target groups that were identified by the LiveGenMon project team were general public, different stakeholders, and policymakers. Within the target group general public, special attention was paid to the dissemination activities for kindergarten children and pupils in primary and secondary schools. Dissemination activities for them should introduce knowledge not only by listening and watching, but also by touching, smelling, and personal experience in the forest. But what is the most effective way to reach children? The answer of the LiveGenMon project team was through activities and teaching materials for kindergartens, schools, and families. 5 different approaches were used to reach this specific target group of kindergarten children and pupils in schools. First, children's books and cartoons about forestry, forests, genetic resources with augmented reality animation as an innovative pedagogical technique. Second, by organizing workshops and public open days for kindergartens and schools aimed at children, their teachers, and educators. Third, by organizing training courses and publishing teaching materials for teachers and educators. In addition, a smartphone app for children, teachers, educators, and parents was developed. And last but not least, organizing events for families. Workshops with active engagement of children were organized in all 3 project countries and abroad, which have reached in total more than 4,000 children. These workshops were organized for kindergarten children and lower classes of primary school children, with special focus on personal experiences in the forest. But we also organized workshops for pupils and students where more complex topics were introduced to them, such as research work, work in the laboratories, forest genetics, and so on. Open days for general public were organized in all project countries. In total, our project team reached more than 4,500 people with activities on the open days. In Slovenia, 9 training courses for teachers were organized in the Slovenian Forestry Institute and the educational group Forest of Experiments in cooperation with the Slovenian network of forest schools and kindergartens. In total, more than 700 teachers and educators were reached. A very successful publication of the teaching materials for outdoor education was the Handbook for Learning and Play in the Forest. It was published in Slovenian in 1,000 copies and in English in 500 copies in 2016. Due to a great interest of teachers and educators, the Slovenian version was reprinted in 2019 in 5,000 copies by the financial support of the Slovenian Ministry of Agriculture, Forestry and Food, and it was distributed to all primary schools in Slovenia. The handbook was also presented to more than 400 teachers at 10 events in bookshops, training courses, project networking workshops, and so on. The handbook is available also online for free download and has more than 11,000 views in each of the languages. The handbook presents 35 entertaining and educative activities for outdoor learning about trees, forest animals, water, and genetic diversity. All activities have been developed and tested in practice by the educational group Forest of Experiments at Slovenian Forestry Institute. The project activities for families have reached more than 2,500 people. In Greece, 6 events were organized for families with 340 family members. In Germany, Walderlebnistag for families was organized with more than 700 family members. In Slovenia, 2 events were organized with the main event, the 7th Križnik Forest Fairy Tales Festival in 2018, which had 1,500 visitors in total. In addition, a smartphone app called Seed Hunter was developed and is available for free for Android devices in Google Play. In this app, the player is searching for the seeds with a smartphone, learning about trees and their life cycle. It motivates players to visit a real forest, park, nature trails, and so on, and connects nature and technology. It also has high educational value since it informs players about the importance of seed collection, rare tree species, and protection of genetic diversity. Three children books and cartoons on forestry were published in 4 languages: English, Slovenian, Greek, and German, a total of 12 books. The first book, entitled Journey to a Big Forest, is aimed at kindergarten children and tells the story of a boy named Simon who meets the 3 characters and their journey to a forest. The second book, entitled Gardens of the Forest, is a continuation of the first book with the story of a girl, Sonia, who helps the 3 characters to save the forest from the bark beetle outbreak and is aimed at younger primary school children. The third book, entitled Forest Diary, is a continuation of the second book and is aimed at secondary school pupils. In addition, 3 short animations with augmented reality upgrade for books were produced, which can be accessed as a free mobile application, Big Forest, in the App Store or Google Play. Through activities and materials, more than 56,600 children, parents, teachers, and educators were reached and informed about the LiveGenMon project and its results. Some of them were reached through more than one approach, which allowed a synergy effect of the dissemination activities. Thank you very much for your attention.
Ursha's presentation, we see how important it is that sound scientific information is transferred to a general public and especially to children who are kind of future of hopefully also science. Are there any questions for Ursha? If not, we will continue with the next successful story about the dissemination, this time about the Greek experience. The presentation will be given by Hrize Sarvani, spreading the word about the LiveGenMon project, Greek dissemination approach. Please.
Good afternoon. We came up with several ideas and ways of spreading the word about the LiveGenMon project. These are some of the most important dissemination events that took place in Greece from the beginning of the project up till now. In December 2015, a Christmas event took place in Thessaloniki. This was our first and probably one of the most remarkable open day events. The idea was to have people write a wish on a card and then place it on a Christmas tree. Small Christmas trees as well as goodie bags were distributed. Our Christmas tree was decorated with cards filled with wishes. We thought that it would be a good idea to expose ourselves to the general public. So, as a LiveGenMon team, we participated 4 times in the Thessaloniki half-night marathon. Different type of dissemination events took place just for families and children. We had the fairy tale day where the LiveGenMon children's book played a major role. The children were fascinated with the stories and the adventures of Simon and his friends. Families and children visited the plots. The youngest children there were 3 years old. We also presented the LiveGenMon project to a whole school consisted of 250 children. Once a year on a Sunday, the Aristotle University opens its doors to the general public. There, we organized a LiveGenMon event where we welcomed children of all ages. We planned games and activities for 3 different age groups. Outdoor, indoor workshops took place, as well as oral presentations for pupils and university students. Probably the most emotional and meaningful presentation was the ones that we gave to special activity groups. From the feedback that we received, we understood that these groups need to feel that they are taken into serious consideration and they're not excluded from the rest of the world. In October 2017, a joint Gen3 LiveGenMon stakeholders event on genetic monitoring was organized in Thessaloniki. The participation was significant and there was an exchange of different approaches and experiences regarding forestry, forest genetics, and dissemination methods. Finally, in our effort to communicate the project, its importance, and its subject, we reached out to important policymakers who have the ability to legislate, make forest genetic monitoring applicable, and improve future forest management. We would sincerely like to thank everyone within the LiveGenMon project for their contribution, as well as everyone who participated in the dissemination activities. Thank you very much for your time and your attention.
Chrissy, thank you very much for your presentation. And I have to agree with Bruno Fadi, who commented that quite impressive communication material. Are there any other comments to this presentation? Or shall we continue with the third LiveGenMon country, Germany? Mark Walter will give us word about their dissemination activities within the LiveGen1 project. His presentation is entitled Sound Science Grounds for General Public Outputs. I'm sorry, my handwriting is horrible. Mark, please.
Hello and welcome to my poster presentation. My name is Mark and I'm responsible for the LiveGen1 dissemination in Germany. Our title is called Tree Faces Forest Genetics at Your Fingertips. Why did we call it that? Well, one of the main challenges in dissemination was that forest genetics is very abstract. You can't really touch it, and it's even more difficult to explain to children. Therefore, we developed special workshops for each target group, like this workshop for kindergarten children, which we called A Forest Has Many Faces. With this workshop, we wanted to promote the basic idea of relationships between trees in a forest. So after talking to them about similarities between them and their siblings and parents, we created imaginary tree families by sculpting faces on the trees with nature materials. All the students had the opportunity to work in our labs and get to know the everyday life of a scientist. But of course, we also did activities on the other main topics like forestry. In the program A Tree for Life, for example, every student planted a marked tree in a local forest, so he or she could visit and watch it grow over the years. On climate change, for example, we organized a breakfast with seasonal and regional food for the children and played games on forests in climate change, which we specifically developed. Speaking of forests and climate change, we invited all the Bavarian schools to apply for an interactive lesson on forests and climate change, where one of us would visit the class in person. The response was really amazing, with almost 250 schools applying for one of the 40 events we planned for earlier this year. Unfortunately, we had to cancel most of the live events due to the Corona crisis, but we will try to catch up with online presentations until the end of the project. I still wanted to mention this because it clearly shows that there's a huge interest from teachers on this topic. But back to the tree faces. We also organized a workshop with local wood carving students, which we called Winners and Losers in Climate Change, where the students had to pick an animal and carve it out of a huge oak trunk. Another perfect link to climate change was that all of the trees we used in this workshop came from a wind throw a couple of weeks before. We presented the sculptures at the Forest Experience Day, which we organized for over 20 institutions that have to do with the forest. Even though it was the worst day of the whole week, we counted over 700 visitors. There was a press tour for politicians and also a competition rally where one lucky visitor could win a balloon trip over Bavarian's most beautiful forests. Last but not least, we also did a lot of professional workshops on forest genetic monitoring for scientists and foresters, including a visit to our plots. Of course, we did a lot of media coverage on all of our activities, many newspaper articles, TV programs on national and regional television. And just now we finished filming a 15 minutes project film, which we are going to publish on YouTube in November. Thank you for your attention. Once again, my name is Mark Walter, and I work at the Bavarian Institute for Forest Genetics.
Mark, thank you very much for this presentation. Since you have another presentation, I suggest that we look at your seed hunter as well, and maybe there will be some comments later on. Please.
welcome to my poster presentation. My name is Mark and I'm responsible for the LiveGEMON dissemination in Germany. In this presentation, I want to talk about Seed Hunter, a forest pedagogic game we developed for Android. In Seed Hunter, the player goes out into nature to hunt for tree seeds. The seeds are over 3,000 virtual objects, which are placed inside forests and parks nearby. As soon as the player approaches a virtual seed, he or she can collect it with a smartphone. Well, so how does it work? As soon as the user opens Seed Hunter, he or she sees his or her location on the map. The map also shows forests and parks nearby where virtual seeds can be collected. What was important for us was to create a game where the player actually has to go into the forest and not just sit on the couch. As soon as the user approaches the virtual seed within 50 meters, the screen changes and the seed can be collected. For each collected seed, the player receives points. Rare points, such as rare seeds, such as elm, for example, give more points than common seeds. If the player finds more than 5 seeds of one kind, the player also receives bonus points for genetic diversity. All collected seeds go right into the heart of the application, the user's vault. By clicking on a seed, the user gets further information on the tree species, its flowering and seeds, the distribution area, and some interesting facts about the species. On this occasion, I would like to thank Euphorgen for letting us use the distribution maps, as well as the beautiful icons for the seeds. As soon as the player decides to create an account, his or her score will be shown in an international ranking list. Of course, you can also play Seed Hunter without registration, but in this case, a competition with other players won't be possible. So what are the benefits of Seed Hunter? Well, for one, like I said, the gameplay takes place outside. The person who visits the most forests wins. Also, visitors can be guided to forests worth seeing, like nature trails or national parks. Furthermore, there's a link between technology and nature with the motto, from smartphone to growing a real tree. In addition to this, we will raise awareness of rare tree species and tree species threatened with extinction. Last but not least, we will transpose the message that collecting valuable seed is an important task to protect genetic diversity in our forests. With the QR code you're seeing right now, you can download it directly from the Google Play Store if you have a code reader installed on your smartphone. Thank you for your attention. Once again, my name is Mark and I work at the Bavarian Institute for Forest Genetics.
Mark, thank you very much for the presentation of the Seed Hunter. I would have a question from my side. Since basically everyone who is hunting for seeds, they are doing this in different locations, and since usually the phones are geolocated, were you planning to collect this information as well as a kind of, let's say, either building up or dancing the map of growing different tree species in the area?
I think this is quite, it's an interesting question, but this is quite difficult to achieve, I think, because it was already very difficult to, because it was so many, it combines so many actors only to get this geo information from the Bayerische Staatsforsten, for example. The communication was quite stressful to get everything in place. So it would be nice, but I think it's not possible, I would say.
I'm sorry to hear this, but congratulations. Anyway, thank you very much. We will continue with the next speaker. The next speaker will be Boris Rantasha. The title of his talk is LiveGEMON Communication as 360-degree approach. Let's roll around. Boris.
Hello today for the second time. My next presentation will be about the whole circle approach to LiveGEMON communication and dissemination. So effective communication of complex biological phenomena needs to be done in accordance to certain findings from research and practice. In this presentation, we will present 7 different tips or guidelines on how to effectively communicate about forest genetic diversity and forest genetic monitoring. So the first guideline is always provide context and help your audience build a basic understanding and see the big picture. So we can start with explaining what are genetics, what is DNA. It depends on the level of knowledge of our stakeholder. And then we slowly move forward to biodiversity and why are forest genetic resources important. So we really need to make sure that our audience understands the basics and understands them well before moving forward. So this pyramid that you've seen several times about the different levels of biodiversity really represents the importance of genetic diversity well. It is one of the main communication tools of the LiveGenMon project. It wasn't really planned in the project. It was made by a researcher who is also an illustrator, Doman Finchgar, and it became one of the basic communication tools of the project. And also this comic represents what is actually being done within the, let's say, field and laboratory part of the project very well. So with these 2 graphics, basically we have the necessary tools to present genetic monitoring to an average Joe. The third point would be that we need to always explain the larger context. So to explain the meaning and importance of forest genetic diversity in the context of climate change and for the future of forests. This way we give our project topic meaning and also entice people to find out more. And the fourth point would be to use comparisons to more familiar topics such as human genetics or edible plant genetics. This is especially very important when talking to children. So this is an example of one of the graphics that we procured within the LiveGenMon project. This is basically represents the genetic diversity of spruce trees and the selection of plus trees. So I think this is quite a good way to show what, let's say, target tree would be. Moving on, we need to use communication tools that are suited to target audiences. So basically for children, we wouldn't use, let's say, longer books. We would go to children's books with interactive components and also cartoons and so on. For policymakers, you need to make your materials short and concise and not give them, let's say, a longer monograph because they probably won't read it. Maybe the most important thing, especially in a longer project, and especially when building solid foundations for changes in forestry practice and policy, would be to engage our target audiences in a continuous dialogue. So it means that maybe every year, every few months, this also works with the media, you call a briefing or a workshop or something and build a relationship towards the topic and also yourself. This is the cover in Slovenian of the Handbook for Learning and Play in the Forest. This is what we used within the LiveGenMon and MANFOL projects to reach teachers, and it was really successful and well received by the target audience. You can download it both in Slovenian and English versions online. And this is one of our children's books titled The Guardians of the Forest, where we explain the meaning of genetic diversity to climate change adaptation in forests. So the final guideline would be to develop relationships. Like we said before, basically good messages and communication will only take you so far. But if you really want to be successful in your communication, in realizing your agenda, then you need to develop relationships and good relationships, both inside and the project team and with the stakeholders. So in LiveGenMon, we did this through many events, through workshops with children, professional events, through events for students, press conferences and media briefings, presentations at fairs, events for teachers, networking events, events for forest owners, different workshops for children, courses for scientists, popular science workshops, and so on. So we will continue to communicate within the Live Systemic project. Hopefully we'll see you on our website and social profiles. And this is it from me for now. Thank you.
Boris, thank you very much for this presentation. The floor is briefly opened for a discussion. If not, then we will move on to our last speaker of the day. Evangelia Avramidou will present us the poster entitled Monitoring Climate Change Adaptation Plans for Urban Green Areas Using Indicators. Live Green Project, a case study in Greece. Since Evangelia is live, I would kindly ask her to log in and give us her presentation.
Yes. Hello, can you hear me?
Yes, loud and clear.
My name is Evangelia Avramidou, and I will present you our new live project. The name is Live Green, promoting...
Evangelia?
Yes?
Just an interruption. Please click on the presentation so that you will have a full screen of your presentation.
Yes.
Right bottom. The last click before the minus.
Yes, yes, yes. Why? Wait, sorry.
Oh.
Okay. Now?
Thank you very much.
Okay. I will talk to you about the Live Green project that Institute of Mediterranean Forest Ecosystems have as a coordinator. The project is about urban green areas and mitigation by using indicators of climate change for mitigation and adaptation to climate change of urban areas. As we already know, cities already face harsh climate conditions due to both obvious anthropogenic impacts, and urban green areas should be considered as vital elements. And now, as we already know, with the new COVID-19 situation, we had all the people had this need to go out and to go to walk in a green park around their home. So it's very vital for us to sustain and include a full plan for urban green areas. Here in Greece, the situation is not good because we don't have so many green areas per citizen. And in Greece, 49% of the population lives in cities of different sizes. The Live Green project aims to incorporate the climate change adaptation and mitigation perspectives, and they will utilize all available tools and indicators at European level in order to assist in strategic planning and management in the context of climate change adaptation and mitigation. So what we did, we have two pilot areas, one is the municipality of Amarousion in Athens and one of Heraklion, Crete. The total budget, the European contribution, and the duration. We have several partners. Institute of Mediterranean Forest Ecosystem is the coordinator. We have a Homotech company, the Central Union of Hellenic Municipalities, Greek municipalities of Amarousion and Heraklion, and the Hellenic Ministry of Environment and Energy. Here you can see the two pilot areas. So we move now that we did some measurements for biodiversity indicators to assess the ecological quality stability of urban green areas. We performed its measurements in both municipalities, and we have biodiversity indicators, which is species richness and sound information index. Then we established two meteorological stations, both in Heraklion, Crete, and in Amarousion, Athens, in order to have the indexes like the physiological equivalent temperature and the predicted mean vote index. And then we have also insects biodiversity indicators, because butterflies in particular are being recognized as a viable environmental indicator. And lastly, we have phenotypic measurements like evaluation of and selection of preferred trees. We had calculation and registration of osmotic characteristics, and also estimation of plant health. From now on, and with Climate Adapt platform, which is the most viable platform for adaptation and mitigation in order to establish a management plan for the urban green areas. We will move to selecting indicators such as tree intensity, tree canopy, and those indicators will have to be the most viable scope of the project is that the indicators will have to establish by a law from the ministry. So each municipality has to sign the Covenant of Mayors, that they have several aspects to it in order to reduce the CO2 emissions, and then to manage all these areas, urban green areas, in order to have some indication before he established the plan, and after he established the management plan of urban green areas. Thank you. I will not have anything else to say because I know it's little time. Thank you very much. Thank you, Troika, for all the organization of the Congress, and hope to see you again soon after this coronavirus situation.
Evangelia, thank you very much for your presentation. I guess people are already a little bit tired of the whole day of lectures, so I'm pretty sure if there are any questions, they will ask you through chat or directly. So, the session 5 is at the end. I would like to thank to all the presenters, to everyone who participated in the discussion and helped to make the half online or hybrid conference more interesting and more dynamic. I would also like to thank to Gregor Božič and Pictorama team for helping with the technical issues and technical support. And of course, thank all of you for participating in today's LiveGenMon conference. Have a nice evening and goodbye.